This method can answer key questions about neutrophil extracellular trap or net formation in dogs or other species. The main advantages of these techniques includes the real-life detection of net formation and identification of net components like myeloperoxidase and citrullinated histone H3.
Begin by obtaining 10 to 20 milliliters of blood from the jugular vein of a healthy canine donor, removing the needle from the syringe, before dispensing the blood into sodium heparin tubes to avoid excessive shearing. Gently invert the tubes a few times to ensure an adequate mixing of the blood and anticoagulant, and check for clots and red cell aggregates before placing the samples on ice. Dilute the anticoagulated blood with an equal volume of ice-cold Delbecco's PBS and transfer 8 milliliters of diluted blood into a 50 milliliter polypropylene conical tube containing 25 milliliters of filtered 3% dextrin. Place the tube upright for 30 minutes at room temperature to allow aggregation and sedimentation of the erythrocytes. Then carefully layer 5 milliliters of leukocyte-rich plasma on top of 5 milliliters of density gradient medium in a 14 milliliter polypropylene round-bottom tube. After density gradient centrifugation, use a 5 milliliter serological pipette to transfer the intermediate granulocyte cell layer into a 50 milliliter conical and lyse any erythrocytes co-aspirated with the polymorph.